Good morning. I'm kind of covering like 2.1, and then like 2.3, 2.4, and 2.5. This is very much a mix and mesh of different topics. So just continuing from yesterday. So 2.1 kind of starts with metabolism. Metabolism. Okay, now metabolism is broken down into two things. There's anabolic and there is catabolic. You might know as catabolic. All right, so basically what we have here is, if I T-chart this, anabolic, this is going to form bonds. This is going to break bonds. This is going to absorb energy. Okay, my shorthand for energy is NRG, so just get used to it. Absorb energy, release energy. So we're making compounds and we are breaking compounds. I am going to do my best to avoid any fancy verbiage. If you want to look up definitions of metabolism, that's fine by me. One way to define it by the syllabus is metabolism is a web of all enzyme catalyzed reactions. I'm going to give you some note-taking skills here. When I write reactions, I'm only going to write R, X, and S. And when I write like energy, I'm not going to write energy. I'm going to write NRG. On the exam, in full, notes helps you out. Okay, I will say this. A key word here is enzyme catalyzed. Okay, so what is metabolism? I hope you all know this. Summation, right? So metabolism is a sum. It's a sum of all the anabolic and catabolic reactions. Okay? It's the sum of all. This is how I remember metabolism. If you add up all the anabolic plus the catabolic, put them all together, and you have either a anabolic reaction or you have a catabolic. Okay? So, now, the next thing about metabolism is it can be linear or cyclic, which means you have step A, step B, step C. Okay? Cyclic looks like a circle. That's it. There are two type of like, okay, this would be metabolic pathways. They can be linear and they can be cyclic. I'll give you examples. Glycolysis. This is glucose, and lysis means split. So glycolysis splits sugar. Okay? So you start with 1 glucose. There are 10 steps. Okay? The whole process is catabolic. Okay? And at the end, you have 2 pyruvate at the end. So 1 glucose is split into 2 pyruvate. There are 10 different steps. Some of those, of the 10 steps, some of those, like, so 1, 2, 3, 4, 5. For example, this is anabolic. This is catabolic. This is anabolic, just for example, right? So if you take those 10 steps, some are anabolic, some are catabolic. In the end, the whole process is catabolic. You're breaking down molecules and you're releasing energy. An example of a cyclic would be Krebs cycle or the Calvin cycle. Now, the Krebs cycle, this is respiration, and this is photosynthesis. So, in the Calvin cycle, we are making glucose from CO2. So, what are we doing? We're taking small molecules like carbon dioxide, and we're making large molecules like glucose. So, is that anabolic or is that catabolic? Well, go back to our definition. We're forming bonds. We're making compounds. So that would be an example of an anabolic. So this would be anabolic. Now, Krebs cycle, we know respiration. We're taking glucose and we're going into CO2, right? That's what we're doing. Glucose turned into CO2. So we're breaking down compounds. So Krebs cycle would be an example of catabolism. Now, both of these process, the Krebs cycle and the Calvin cycle, both of them are cyclic. They're circles, A to B to C, and they go back into A. Whereas, like glycolysis is linear. They give a real question on the exam might just be a picture of a pathway you've never seen, and the question is simply: is it linear or is it cyclic? Are reactions anabolic or are they catabolic? Okay, now that is metabolism. Now for there is a thing about inhibitors. So inhibitors, what does it mean to inhibit? It means to slow things down, right? So it slows things down. So there's competitive, non-competitive inhibitors, and there is one more type. You know what? This is HL. We will come back to this. So HL, there's a bit about inhibitors, but I hope everybody knows the word inhibit. Inhibit means to slow things down. So inhibitors can slow down a pathway, okay? Now, this is obviously connected to enzymes. Enzymes we will cover another time. So where am I going from here? I'm going to keep with our lesson from yesterday. So all life is made of HONC, honk. Now we have 4 molecules. We have carbs, we have lipids, we have nucleic acid, and we have protein. Okay? Carbohydrates are CHO into 1, 2, 1. Lipids are also made of CHO. Nucleic acid is CHON. Sorry, P. CHONP. And protein is CHONS. Okay. Now, remember that. Okay, that's from last week. The next thing we're going from here about molecules is we're going into monomers and polymers. Okay, so make another chart over here. Now, we have nucleic acid, we have protein, and we have sugar. No lipids. There's no monomer or polymer for lipid. Okay? So, nucleic acid, these are called nucleotides. You know these. DNA, this is A, T, C, G, and in RNA, there's U. So, what do we call a nucleotide that's a polymer? We say polynucleotide. Okay? Quick aside, you may see a few things. On exam, you may see this. You may see nucleotide. You may see oligonucleotide. And you may see polynucleotide. Nucleotide is one. This is some. This is many. So an oligonucleotide is smaller than a polynucleotide. So an oligonucleotide, for example, could simply be 10 or 20. Now, we may use you'll also see when they say nucleotide, they might also say base pair. So BAS pair. Now, if you remember your DNA, A pairs to T, C pairs to G. So if I say a molecule has 20 base pairs, that means there's 1, 2, 3, 4, 5, 6, there's 20 nucleotides. It's not 40. Okay, so nucleotide, oligonucleotide's bigger, polynucleotides even is bigger. Okay, proteins have two names. They're both, you can use both. We say amino acid or you say peptide. Both are okay, both are acceptable, no both. So generally what we say, okay, so we have, that's the monomer is amino acid. Now the polymer is a polypeptide. Now, a polypeptide does not mean protein. So, if I erase all this, okay. So, a protein, so if it's a protein can be many polypeptide. So, for example, hemoglobin. Hemoglobin, it's a protein. What does it do? It carries oxygen in the blood. It's made of 4 polypeptide. Now, one protein can be one polypeptide, or a protein can be made of many polypeptide. A polypeptide just means many amino acids. So, the monomer, I should write this neater, monomers, okay, is amino acid or peptide. A polymer is a polypeptide. A polypeptide does not mean it's a full protein. It can be. A polypeptide can be protein, but it doesn't necessarily mean, like a protein can be made of more than one polypeptide. A sugar Okay, now sugar, there's a little bit more for sugar to know. Okay, so the first thing about sugar is that the monomer is okay, I'm going to go down here. I'm going to just shoot over here. Okay, so we have saccharide. Saccharide means sugar. So there is monosaccharide. Okay. We have disaccharide. And we have polysaccharide. We will go more in detail on this later, but everything I'm saying right now is just pure memorization. There are 3 monosaccharides. There is glucose, galactose, and fructose. All of these are C6H12O6. So we have glucose, galactose, fructose. Disaccharides, you have maltose, sucrose, and fructose. Lactose. All of these have glucose. So maltose is glucose plus glucose. Sucrose is glucose plus fructose. And lactose is glucose plus galactose. Okay? So, glucose, galactose, fructose are your monosaccharides. They form maltose, sucrose, lactose. So, maltose is glucose and glucose. Sucrose is glucose and fructose, and lactose is glucose and galactose. Now, the polysaccharides, there is cellulose, there is glycogen, there is starch, and chitin. Within there is also amylose and amylopectin. Amylose is cellulose is amylose or amylopectin. Okay? Amylose is alpha D glucose, and there's 1,4 bonds. And amylopectin is going to be branched. So amylopectin is also alpha D glucose. Now everything we draw is D. We don't draw L. L is the mirror image. So amylopectin is alpha D glucose, and it's 1,4 bonds and 1,6 bonds. Now, you're probably asking, what is 1,4,1,6? We did not cover that. Yes, I did. If you draw glucose, CH2OH. OH. 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 1, 2, 3, 4, 5, 6. If I draw another glucose, remember I don't need the H's, so OH, 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 CH2OH. Now remember, look, I'm counting 1, 2, 3, 4, 5, 6. O's, there's one, check the O's, 1, 2, 3, 4, 5, 6. I can count the H's, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Right? So I can count all the O's, the H's, they all have a number of bonds. So here's my number 1, here's my number 4. So there's my OH, my water goes away, and I have a 1,4 bond. So here's my glucose molecule. Now see, the water has gone away. There's only one O left, which is now here. Sound effects help you to memorize this. Oopsie. Okay. Bam! Look at that! This is a disaccharide. There's my disaccharide. Okay? You have to be able to do this from memory. So this is a 1,4 bond. This is a 1,4. Right? If I form a bond between the 1 and the 6, if I go from there, that's a 1,6 bond. 1,6 bond creates a branch. So like if I was to draw a 1,6 bond, I would have one molecule here, and I'm going to draw really, so the other one would be down here. Right, there's my C, and there's my bond. 1, there's my 6, there's my 1, there's my 1,6 bond. Okay, so amylose, amylopectin. Shoot, oopsie, not cellulose, amylose, amylopectin. Uh-oh, too bad I can't fix that. Amylose is our starch. Glycogen is completely branched. So if you look at glycogen, it's just like a web. This is glycogen. Okay? Chitin, this is shells, nails, okay? Cellulose, we know this is in cell wall. We'll get more into this, but look, this is important. This is kind of what I'm talking to you about today. So this whole lesson today is really about how we form. So this is a two sugars come together. You got to be able to draw this out. Okay? So this is my bridge. So we did polysaccharides, right? There's your sugars, your saccharides. So we did sugar. We do not need to do this. When we do DNA, and when we get to the DNA topic, we'll spend more time. So for now, we're done. We are literally done with DNA. All you have to do for DNA, for right now, here's your phosphate group, 5 carbon sugar, nitrogen base. That's it. That's all we have to know for right now, right? If you really want to know why we say 5 carbon sugar, because we have DNA and we have RNA, right? The DNA bases are ATCG and RNA is UACG. That's it. More detail coming later. You do not need to draw this for nucleotides. So let's go to protein. Okay, so drawing an amino acid. So we have N, C, R, H, C, O, O, H. Okay, and I'm going to draw this out completely. All right, that's my peptide. Okay, my next peptide, I'm going to draw another peptide. So N, H, H, C, R2, we'll call this R1, H, C, double bond O, O, H. There's my water. So they come together. So we have N, H, H. This is called the amine group. C, R2, H, C, N, H, C, R1, H, C, O, O, H. This is called carboxyl. This is called a peptide bond. Small note, this is the correct way. Your O goes up and your NH goes down. If you draw them, you'll see them done incorrectly on the web. If they both go up, you're not wrong. You're not going to get docked points if you draw it wrong. But just so you know, if you want to be absolutely correct, O goes up, NH goes down. This is a 3-point question. Drawing the synthesis of a dipeptide. Okay, so remember how we said we had mono, di, okay, so here's the mono, di, oligo, poly. So, and actually you can also do tri. I guess we could do tri as well. So, mono is 1, di is 2, tri is 3, oligo is some, and poly is many. Okay, so this is a dipeptide. Okay, 3-point question. The first point would be drawing this correct. That's the first point. The second point is drawing water. You must show water leaves. If you're drawing the synthesis of a carbohydrate or a protein, you must draw the water is leaving. So you must draw with an arrow. You must write H2O and write water leaves. And you guys circle the water. If you make a mistake, if you circle these two, you're probably not going to get marked down. You probably won't get marked down if you pick the wrong one. But it's H2O, right? What's water? We should all know this. H2O. If you don't circle 2 H's and an O, you're wrong. If you circle the OH and the R group, you're wrong. If you circle this O and this, like if you go there, that's wrong. OH and H. That's it. Okay? So water. That's the second point. The third point is the correct dipeptide. The same goes for drawing a carbohydrate. So if I'm drawing the synthesis of a disaccharide, 1 point for drawing the correct sugar, 1 point for showing the removal of water, and 1 point for showing the disaccharide. Okay. Dehydration synthesis. What's happening is in the word. Dehydration synthesis. Remove H2O and make compound. Okay? This is also called condensation. Okay? Now, the reverse of this is known as hydrolysis. So this is add H2O split. So the synthesis of a dipeptide, this is a dipeptide synthesis. I made a dipeptide. I removed water. Dehydration synthesis. My carbohydrate. I removed water. Dehydration synthesis. Now remember, lipids have no monomers. So, if I'm doing now this is completely bonus. They could ask it, don't think they will. C, OH, CH. Now, this is glycerol. Now, I take 3 fatty acids. Now, for sake of speed, I'm making all of these saturated. So CH2 and CH3, HOC. You can also draw it like this. This is the same as this. CH2N is this. There's a C, there's an H, there's an H. And this is CH, right? H. So CH3, CH3. I'm writing, so this part, ignore. It's all the same. So look, there's my OH, there's my OH, there's my OH. You must draw the water. Woo, woo, woo. So we have 3 times H2O. Now comes the synthesis. So this is, what am I doing here? The dehydration synthesis of a triglyceride. Okay, so step 1, I drew my step 1, fatty acids and my glycerol. Step 2, I remove the water. Step 3, the final product. So these are called ester bonds. So starting with my glycerol, O, C, double bond O. Remember, only that original O is left. Right? This is gone. And I'm just going to draw it out. There we go. I have 1 fatty acid, 2 fatty acid, 3 fatty acid. 3 fatty acids make a triglyceride. There's my glycerol. People, we're halfway done. Woohoo! Yay! Halfway done. Okay. No, we're not. We're almost done. And I'm just going to draw two things in reverse. So that was all dehydration synthesis. Now, let's just do hydrolysis. So, I'm going to draw my dipeptide. NH, there's my amine group. Carbon, H. I'm going to draw it out this time completely. C, N, H, C. Because they might do it either way. They could do it either way. They can call this an R group or not. H, C. So I'm messing with you. This is a different R group. See, there's one R group and there's the other R group, right? Here's my dipeptide. So that's 1 point. That's step number 1. That's 1 point. Number 2. Water goes in. You must draw the water going into the bond. Step 3. Just draw them separate now. I hope you notice what I did. This is the same as this. See, there's my CH3. CH3. So if you draw it out, it's fine. If I use R groups, if I just screw that and I just wrote R1, R2, R1, R2. This is also correct. Okay, that's step number 3. This is the hydrolysis of what? What did I draw? Hydrolysis of a dipeptide. So now, let's do the hydrolysis of a carbohydrate. So, if you draw some H's like I do on accident, it's fine. CH2OH. I saw some of you not connect this. Make sure this C goes to this C. Don't do that. That's just wrong. Okay? Do not draw to the wrong thing. Okay? Okay. H. If it helps draw it out, you can. OH. Okay, so that's step number 1. Step number 2, draw the water going into the bond. And then step number 3, draw them separate. There we go. Those are my Hydrolysis is metabolism, is anabolic or catabolic. So hydrolysis, we are breaking compounds. We're breaking, so this hydrolysis is catabolic, and dehydration synthesis is anabolic, right? Synthesis, this is anabolic. Okay. Monomers, polymers, honk rule. We're going all the way back to where we started. So, at the end of this lesson, first, this is what you can, after watching this video, you can do this. You can define metabolism. Metabolism. 2, you can distinguish catabolism from anabolism. Guys, I know I'm not great with this. I'm just lazy, but I say these words all the time. Catabolism and anabolism. Distinguish means make clear the difference. If I show you a reaction, you can tell me if it's anabolic or catabolic. Define metabolism. Same with distinguish linear from cyclic. If I show you A, B, C, if it's a straight line, it's linear. If it's a circle, it's cyclic. Okay. The next thing you can do from today's lesson. Over here. Let's go over here. Number 3, monomers, polymers. This is make vocabulary. Mononucleotide, one nucleotide, dinucleotide, trinucleotide, oligonucleotide, polynucleotide. Okay, so you have like nucleotide. Okay, it's already in the thing. Monomers, polymers. What is the monomer to the polymer? A nucleotide makes up nucleic acid. Amino acids make up protein. Peptides make up polypeptides. Monosaccharides make disaccharides. Disaccharides make polysaccharides, and so on. Okay, the last thing you can do is you can draw, this is the hard part, drawing hydrolysis and condensation. Sorry guys. Draw hydrolysis and draw condensation for a disaccharide and a dipeptide. So, can you do the hydrolysis of a disaccharide and a dipeptide? Can you do the condensation of a disaccharide and a dipeptide? That's 4 different things you need to draw out. 4. If you can do triglyceride, you, sir, are a pro. If you can do triglyceride, this is bonus. Okay? No monomers for lipids. You do not need to draw this for a nucleotide. Just for disaccharides, dipeptides, and triglyceride is bonus. 45 minutes. That's a one long video. I am done. I hope you are too.